Hello, everyone. Thank you for joining us for today's webinar. My name is Agata Sobirajevich, and I'm a team lead within the NGS Study Management team at Azenta Life Sciences. At Azenta, we are here to help clear the path for researchers and life science companies to accelerate scientific breakthroughs. As a global leader and full service provider, we offer a comprehensive genomics and analytical services portfolio including Sanger DNA sequencing, next generation sequencing, gene synthesis, molecular biology, bioinformatics, and GLP regulatory services. Now I would like to introduce our presenter, Dr. Mato Lagator, and the webinar Understanding Biological Mechanisms to Better Predict the Evolution of Antibiotic Resistance. WHO recently declared that antimicrobial resistance is one of the top 10 global public health threats facing humanity. Researchers like Dr. Lagator are using synthetic biology to experimentally influence evolution by manipulating bacterial gene regulatory networks. Mato carried out his undergraduate work at Harvard University and his PhD at the University of Warwick. Mato joined Manchester University to start his own research group as a presidential fellow and has since transitioned into Wellcome Trust Royal Society Sir Henry Dale Fellowship. His research group investigates how the mechanisms of gene regulation in bacteria shape the evolution of gene regulatory networks and applies this knowledge to tackle the problem of antimicrobial resistance. Throughout the event, please feel free to send any questions you may have through the question chat box located on the right-hand side of your screen. Mato will answer your questions during the dedicated Q&A session. And without any further ado, here is Dr. Mato Lagator.
Thank you so much, Agata. Thanks everyone for joining. Outside of those complicated English-based position definitions like Wellcome Trust Royal Society Sir Henry Dale Fellow, for all intents and purposes, I'm a group leader, and I think that's the easiest way to summarize what I do here in Manchester since starting some years ago. Yeah, so what I wanted to tell you about today is really kind of first to give you a sense of how we approach studying evolution in a way that, for reasons that are still unclear to me, is not necessarily done that often, and then how such approaches can actually help us do and understand evolution differently and understand in a way that we can start predicting some of the key aspects of the evolutionary process that are not normally that are typically very unpredictable. But I'll build towards all of that, and I want to take you on that little trip, a philosophical journey at first. Sorry, my mouse was not here. We are. So same title, different intro page. I just want to introduce my beautiful graphics here. So let me first start by just very broadly, in a very naive way, talking about why do we even care to study evolution? And on one hand, it's because it's really awesome. I mean, life is really cool and incredibly diverse and has led to so many different forms and shapes and sizes of creatures that live here that it's really cool to understand the rules that led to all of this diversity that we observe around us. On the other hand, and Agata already sort of hinted at this, evolution is maybe today more relevant to our lives than it ever was, at least in the recent human history, and that's the evolution of resistance to various pesticides and antibiotics, which are really affecting our ability to feed ourselves and also to survive and to the age that we are currently accustomed to living comfortably. And both of these, food security and health through the risk and minimization of the available antibiotics pose really large risks to the humanity. And the kind of work that we do wants to link the basic evolutionary properties to how they affect these applied evolutionary questions, let's say. So to give you a sense of what we are, kind of the gap in knowledge that we are trying to address is, I want to again, very illustratively and broadly talk about the field of evolutionary biology historically. And in some ways, the field of evolution is really well mathematically understood, particularly some aspects of evolutionary biology. And this is largely to do with the fields of population and quantitative genetics, which for the last 100-ish years have developed a solid mathematical understanding and good mathematical tools for predicting how a population changes over time under selection. But to do that, these approaches have to assume some really critical properties of systems. And in particular, they have to assume what are the effects of mutations. So we have a diversity. Once we know what the diversity is that's present in a population is, we can model with relative accuracy how that population will change under given selective pressure. But we don't currently have very good tools and a good understanding of where the diversity comes from. We know it comes from mutations, but we don't, in vast majority of instances, we take any new mutation and say, the defect of that mutation is random. We draw it from some assumed distribution or do something like this. And the reason why we are sort of not very good at predicting what mutations do is because to a large extent, our approach to understanding evolution, understanding what mutations do is experimental and observational. And what I mean by that is not an insult to the fields that I also belong to and I have done a lot of work in, but really just it's a way of looking at evolution. We have something that evolved, we sequence it and we find out what mutations are responsible and we sort of stop there. And I'm going to use this analogy in an example that I'm going to do about in a bit. But the problem with this approach is that because we are victims to a specific outcome of evolution and looking at what mutations gave rise to that specific outcome, it becomes a bit difficult to generalize beyond the particular system we are studying, because we don't know the rules, the mechanisms, and how they relate to the specific effect of mutation that we observe. On the other hand, I want to highlight some of the aspects of the field of molecular biology, which is probably the second largest field in biology if you take kind of evolutionary biology and molecular biology as big fields. And this field has been obviously exceptional at providing extensive and detailed descriptions of a lot of biological systems, but almost always those systems are studied in isolation. So when you're doing some molecular biology work, you don't want to change 50 systems and try to figure out what happened. You try to really change only one system that you're interested in and you ignore everything else around it. And importantly, molecular biology most of the time is studied outside of the evolutionary, the ever-changing and temporal context in which all biology actually happens. We try to keep our systems as non-mutating as possible to understand how they work. And so really the overarching goal and vision for the research that's now conducted in my group is to link these two fields that are largely disparate, to link the fields of evolutionary biology to the field of molecular biology. And to do that, we borrow a lot of tools and techniques and approaches from synthetic biology. And we use a lot of really interdisciplinary approaches. And for a while, I felt cheeky using this as the way of just like self-advertisement because it's a nice word to use. But I realize now that I'm in a position to kind of translate this knowledge to others, I realize that we really do combine a lot of really different fields together, the three that I mentioned here, but we'll use a lot of population genetics in our work. We use a lot of biophysics, combine them with molecular biology, combine them with mechanistic modeling. So we really do interdisciplinary research. I'm saying this because I always feel a bit uncomfortable advertising myself like this, but I do think it's true. And so we do this in order to understand how the existing mechanisms in biological cells, we study bacteria, so in individual organisms, how they constrain evolution. We look at how the way in which a system works defines the way in which that system can change. And for whatever reason, this is not often done in evolutionary biology, even though it, to me, seems pretty obvious. And we do this because it allows us to generalize beyond the specific system we're studying, because now we're understanding the rules rather than the examples of what we are of the system that we are looking at. And that then allows us, hopefully, to predict evolution. So that's a very broad, philosophical, if you will, way. And I want to give you a couple of examples of how we applied this up to this point and where we are taking. And then at the end, I will tell you a bit about where we're going in the future. So when we're focusing on what mutations do, we specifically in my group in the past, and this is my own work to a large extent, have focused on promoters. And the reason why we care about promoters largely stems from these two French gentlemen, Jacob and Monod, who discovered the lac repressor in bacteria, which was really the way of understanding that cells are not just making all the genes all the time, but rather they're coordinating expression of genes within the cell. And this coordination happens in bacteria, in particularly at the pieces of DNA that are called promoters, which is where all of these transcription factors bind to integrate the signal and produce a desired or evolved outcome. And these systems end up being so important that they form these really complex gene regulatory networks. This is an example of gene regulatory network illustrated for E. coli. So there's a lot of transcription factors coordinating and communicating when and what time and in what environment to express which gene. But our approach is not to focus on large systems like so much of systems biology as a field has done, but really to go other way around and try to isolate individual systems and figure out how they work. And to do that, we use a very, very simple system. We have some gene here shown in this white arrow, and we have some promoter. These are not to proportion. Genes are generally much longer than promoters. So we have some promoter that we put in front, and this promoter in the systems that we play with can have two different states or two different types. One is a constitutive promoter, which is a promoter that really only binds, as far as we know, RNA polymerase associated with a given transcription, with a given sigma factor. Right? So this is a constitutive promoter. On the other hand, that promoter can be regulated, which means that it binds RNA polymerase, but it also potentially binds one or more transcription factors. In this case, I'm just showing an example of a fictional repressor here. All right? So promoters are really important, and what we focus on is the first part of this talk. I want to focus on the constitutive promoters. And constitutive promoters bind RNA polymerase, as I said, in combination with the help of a specific sigma factor. And there is a whole bunch of sigma factors that are associated with different, some of them are the housekeeping sigma factor, like the sigma 70, and the other ones are associated with specific environments or conditions where you need to express a whole host of genes. And so in this study, we focused on RNA polymerase sigma 70 complex and how it interacts, trying to understand how the mutations in the promoters, in constitutive promoters, affect the binding of RNA polymerase and hence the expression of the downstream gene. So let me give you a bit of an example of how we actually go about studying promoters. So first I'm just here outlining our very loose experimental procedure. So first we experimentally probe synthetic promoters. So we make simple synthetic systems where we control as many parameters within that system as we can, so that when we change something, we have a better hope of understanding what the consequences of that change are. Then we try to understand their underlying mechanisms that governs, in this case, how promoter functions. And then we try to develop predictive models based on that mechanistic understanding. We try to model those mechanisms of how promoters function in order to develop a predictive model that can predict what mutations in that system do. And then we explore the consequences of those models to study evolution. So to give you an example of a couple of studies where we did this and how we iteratively build our understanding and our ability to predict the effects of mutations in bacteria, I'm gonna tell you about a very specific promoter, which is a constitutive promoter. It has a very specific sequence, and it's what I'm gonna call the standard view of constitutive bacterial promoters, which is that RNA polymerase comes and binds to a specific binding site. If this is DNA, RNA polymerase comes and binds specifically on that site, as you can see here. And it's always bound to that site. So mutation just describes the binding to that specific location, right? And I'm going to introduce this as a standard view of bacterial promoters because that is how vast majority of papers think about promoters. So what did we do? What did we do to study mutations in these promoters? Well, first, as I said, we experimentally probed the system. And in this particular study, what we were really interested in is the interactions between mutations. We're interested in how single point mutations, when you have one mutation and another mutation, do they interact with each other? And this is something called epistasis, which is a very notoriously complicated and sort of esoteric and mystical property that plagues a lot of evolutionary biology. And it's because we don't know its origins. We measure epistasis in the lab and we say these two mutations interact in a specific way, but we very rarely know why they interact the way they do. And knowing this is actually, knowing the nature of epistasis between point mutations is incredibly important because it is what defines the structure of the landscape along which the population can evolve. So some types of epistatic interactions, often lack of epistasis between mutations, gives rise to simple landscapes like the one I'm showing here, where a mutation in red can very easily find its global optimum, right? The highest points of this landscape. And this is a fictional two-dimensional genotypic space. Obviously, this is a highly multidimensional space in reality. But different types of epistatic interactions between mutations can give rise to a more complex, multi-peaked landscape where a mutation here shown in red is going to find the local peak, but it's actually going to struggle finding the global optimum because once it climbed the local peak, it needs to climb down, which means reduce its fitness in order to climb back up to the global optimum. And just to give you a more specific quantitative definition of epistasis that I'm going to use here is we consider, because we study gene expression, and we have a wild type which has a given gene expression level. And that wild type can be mutated with single permutations A and B. This is, again, just a fictional example. And both of these mutations here increase gene expression level by a certain amount. So then the question is, what is the gene expression level of the double mutant? And the way that epistasis is defined is that we have a double mutant in case it's in case it's not, if the two mutations are not epistatically interacting, the double mutant is just the effect of A plus the effect of B. So it's an additive assumption that we have here that A plus B equals the double mutant. Now, of course, if the two mutations interact, they can have they can be in positive epistasis, which here means that their double mutant, the measured double mutant effect, is higher than what you predict based on individual mutant effects. And it can also be negative epistasis, where the double mutant expression level is lower than what you predict based on individual mutant effects. And so this is just a definition of epistasis because it's relevant to understand. There's other types of epistasis that are very important, but here we just focused on what's called magnitude epistasis, which is really just changes in the magnitude of the effect. Is it more fluorescent or less? Is the gene expression higher or lower than what you predict based on single mutant effects? So now back to our little promoter system, just to tell you, in the lab, we make this very simple plasmid system that has a yellow fluorescence marker that's controlled by promoter and put on a very small copy number plasmid that has only 2 to 3 copies. And this allows us to introduce mutations into this promoter and study the effects of mutations on epistasis. And so let's think about how people typically study epistasis, because I want to illustrate back on that kind of philosophical point I was making earlier. So what we did to study epistasis and what would be typically done is we would create a bunch of mutants. In this case, we made 240 random double mutants and all of their corresponding single mutations in the promoter so that we can measure the effects of single mutation A, single mutation B, and their double mutant AB. Then we measured their effects, as I just said, and then using those measurements, we calculate epistasis, and we get some description of epistasis. Excuse me, just gonna have some water. So here we have some description of the system, and typically most studies of epistasis, this is what they report. There is some interactions, here is what they are. And what you're looking at here is at the x-axis, the additive prediction, which is the what we predict based on individual mutant effects. And then here is the actual measurement of the double mutant. Any deviation below the line, the diagonal line, sorry, that's below means that the system is in negative epistasis. And if the double mutant measurement is above the horizontal, the diagonal line, it means it's in positive epistasis. So what we then wanted to do is actually try to understand where this epistatic interactions come from. And the first kind of hint that something maybe interesting is happening here is we actually took account of the double mutants in positive and negative epistasis. And what you're seeing here is that there is many more mutants in negative than in positive epistasis, and the darker color just indicate those that are significantly different from the null model prediction, while the ones in positive epistasis are much less frequent. So why is there this difference? Why is some much more frequent than others? And to do this, we actually looked at some biophysics that govern how molecules bind to each other. And this is whooping 100-something-year-old physics developed by Boltzmann and other physicists at the turn of the century. And to do this is that to describe what I mean by this is that we model the binding between RNA polymerase with a sigma 70 factor to DNA using the simplest possible assumption, which is that each position on the binding site is independent and independently contributes to the energy of binding between the two molecules, right? So we use this biophysics of protein DNA binding, and we filter it through what I'm going to call a thermodynamic model in gene regulation. And the way this model works is that it says, well, okay, in your particular system, in the particular system you're studying, Mato, there's two possible states. One, that the promoter can be unoccupied, nothing is bound to it, in which case there's no transcription. The other, that RNA polymerase is bound to the promoter, in which case you have transcription. And using the energy of binding between RNA polymerase with sigma 70 to the given variant of the promoter, we can calculate the probability of the system being in this on state. And again, this is 100-year-old physics. And then we just say that this probability of being in this state is somehow directly related to gene expression level, right? So gene expression level, we assume, is proportional to the probability of a system being in the on state. And what's really interesting and cool is that this relationship between binding energy and gene expression level, or the probability, is not linear like shown here, but it's actually a curved sigmoid relationship, but here I'm showing the biologically relevant part, which is curved in this manner. And the fact that this is curved gives us some really cool ways of predicting epistasis, which were not possible before, without realizing this, what I would consider pretty obvious and straightforward. We're just considering how two molecules bind to each other. We're not doing something crazy fancy here. So to illustrate what I mean is consider now back to our definition of epistasis. I'm going to draw it here a bit differently. You have a specific wild type, which has a specific energy of binding between polymerase and promoter, and that gives specific gene expression level. And then you introduce two mutations, which increase the binding energy. And of course, in physics, increasing energy means you bind less. So now you decrease the gene expression level because these two mutants, promoter mutants, bind RNA polymerase a bit worse. And so what should be the double mutant effect? Well, following our linear additive model, it should be on the line that linearly fits A plus B. The effect should be double mutant here. But that's not what happens. The double mutant has to be on this line because that line is defined by physics. So by definition, we have a double mutant, which is in negative epistasis. Its gene expression level is lower than what you predict based on the energies of binding of individual molecules. And what's really cool here is that because this line is curved in a consistent way, in a monotonic way, we can get very specific predictions of epistasis based only on the sign of individual mutation effect. So we only need to know if mutation A negatively or positively affects expression, and if mutation B does the same, and then we can predict if the double mutant will be in positive or negative epistasis. And it's very consistent. And this actually is really cool because, as I said earlier, predictions of epistasis are extremely rare. And here we can predict sign of epistasis only if you know what the single mutants do, and you don't even have to know the magnitude. And this actually illustratively explains what we saw in our data. And if you remember this, well, I haven't said this before, but the promoter we use is the lambda PR promoter, which we mutate. And this lambda PR promoter in its wild type state, it's a very strong promoter. So it has a very strong expression. So most mutations have a negative effect on expression. So most double mutants are in this minus scenario, which is why they're in negative epistasis. So as I said, this allowed us to predict epistasis based only on the individual mutation effect, something that's been really difficult to do before. And we do validations of this model in the study that you can find in the paper. And what I should have mentioned is a part of our experience with the Zenta is precisely to give us really reliable and rapid sequencing of these large mutant libraries that we're making. But in the second study, which I'm building towards now, I'm going to tell you even more about large-scale sequencing and how it can be utilized to make these models even more predictive. And it starts by recognizing a problem with the specific model that I told you, which is this standard view of constitutive bacterial promoters. Something's a bit off with it. And specifically what is off with it is that when we take a given sequence, like the lambda PR promoter, lambda is a bacteriophage, by the way, I should have said, and PR promoter is a common promoter found in lambda bacteriophage, which is a really well-studied promoter, much like the lac promoter and things like this. So we mutate this promoter, and when we create a mutant library around that promoter and develop a thermodynamic-based model, we get very good predictions. You know, these are pretty reasonable R-squared values of 0.7, 0.8. But when we then apply that same model to random sequences and try to predict the expression level from those random sequences, we get pretty poor predictability of 0.4, around R-squared of 0.4. And what I'm going to tell you now is how we can learn more about the way in which system works in order to improve our predictability of that system. So the question we asked ourselves is maybe the reason why this model is not working well that we have an unrealistic view of how promoters work? We take the standard view where one molecule of transcription factor, in this case RNA polymerase, binds a specific location, we only mutate there, whatever happens around it, nobody cares. Maybe there's a problem with that model. Maybe that's not how RNA polymerase really works in reality. So to get deeper into this, what we did is we repeated our process of probing a synthetic system, understanding the mechanisms that govern it, developing a different model, and then exploring the consequences of that model. So what is the system we explore here? Well, actually, it's the exact same system that I just told you about, promoter that controls the expression of a yellow fluorescence marker, and this is the lambda PR promoter. And then what we do is we introduce a lot of random mutations into this promoter. We mutated with with 12% mutation chance at every position in the promoter. This promoter is about 80 base pairs long, so each position in those 80 base pairs has 12% chance of not being the wild type. So being 4% chance of A, C, or T if the wild type is G. So we create this large mutant library, and then we do what's called the sort-seek experiment, which is that we take this random mutant library and we split it based on fluorescence levels. You use FACS, fluorescent activated cell sorting, to split this library into mutants that have low expression, that have no expression, low expression, intermediate, and high expression, corresponding to different shades of yellow here, because we use a yellow fluorescence marker. And we split this library, and then we PCR tag all together. At this point, we still don't know which mutant corresponds to which fluorescence level or which expression level. We just sequence tag all the mutants in library 1 with, in this case, green tag, and library 2 blue, purple, and red, respectively, as you can see here. And then we send it off to our good friends at the Zenta, who sequence all of this for us and send us back this huge data set of thousands of sequences that are tagged, sequencing with a high coverage. So then we consider only sequences that have at least 30x coverage, because we realized why not? We don't have to be conservative with our data. We can afford to be conservative with our data set. And then based on each one, we can reconstruct the expression level of that mutant by looking at the distribution across the bins, right? And what we end up with is the mapping between individual mutant sequence and expression level. Sorry, somebody's just entering my office here. I apologize for that. Because I share an office and there's some movement. So we can now reconstruct and have a mapping between promoter sequence and its gene expression level, right? And this is the process and how we do that. So that's called sort-seq experiment. And what we do here is we mutate around a lambda PR promoter. So if you imagine this as an entire possible genotypic space in some fictional two-dimensional rectangle, we only sequence about around the specific mutant, around the specific wild type with a 12% mutation rate, which is this lambda wild type PR promoter. Okay, so that's the data set. Now we want to understand the mechanisms that govern how this promoter works. And to do that, we consider one of the mechanisms of how promoter works is this really detailed biophysics that determines the energy of binding. I told you about why we want that in the previous part of the talk, but really what that means, it looks something like this. This is what's called an energy matrix, and you can derive that using some combination of machine learning and Markov chain Monte Carlo derivations. You can use that large data set that I just described to you, and you can derive how every position in the binding site of RNA polymerase affect the energy of binding between DNA and polymerase. And that's one component to what we can derive, but this was known. This allows us to still have that old way of, or standard way of looking at what promoters are and how they function. One position, polymerase binds to one position, now we can calculate how the energy of each mutant in that position changes. What then we also did is, if you consider polymerase as having two binding domains, the -10 and -35 elements, as they're often referred to, there is some spacer between them. And what we considered is that, and this was known before, that the spacer can vary, but what we managed to calculate is the energy penalty of varying this spacer length. And we could also do that from the same data set we had before. Another thing we did is we considered multiple binding configuration for polymerase, because if you consider the promoter to be this whole sequence that I'm showing here, polymerase doesn't have to bind just to this position to transcribe this YFP. It can bind a bit more upstream, a bit downstream. All of those transcripts are going to have a fully functional version of YFP. So it doesn't matter that polymerase binds to a specific location. It can move around along the promoter and bind to different locations. And wherever it binds, as long as it produces a viable transcript of the gene that we're measuring, which is YFP in this case, yellow fluorescence protein, that's a viable transcript. So polymerase doesn't have to have a specific binding site. It can bind anywhere. And then the question is that binding productive? Because, for example, polymerase can bind a bit too close to the ribosomal binding site, in which case the transcriptional start site would miss the ribosomal binding site, which means that that's not a viable transcript. So we can account for this kind of mechanism of promoter function as well. Finally, we can also look at the rates at which, we can look at interactions between positions. So individual, I told you, independently position, let's say second position here, can have some energy value, but that can interact with some weird position number 32 in this particular illustration here, and they can have some dinucleotide interaction. So these don't have to be independent of each other. And finally, we also look at the fact that the energy of binding between RNA polymerase and DNA can also influence how quickly RNA polymerase leaves the promoter. And this is important because RNA polymerase's job is not just to bind like any other transcription factor, it also needs to leave the promoter. So it's possible that if it binds a promoter more strongly, it's going to leave it more slowly, which actually at some point then reduces the level of transcription. So now we have all of these different mechanisms, different ways of thinking about how polymerase works compared to that standard model that I described before. So what we then next did is try to incorporate it into that statistical thermodynamic model that I described to you before, which relates binding energy to the expression level using some sigmoid. Like previously, I've only shown you up to about half of this curve because that was relevant for the system we're studying. So we can calculate this binding energy using the energy matrix that we have. We can incorporate all of those different aspects of how promoters function that I just described to you. And what we can do then, and we can train all of this model based on this very specific library that is very local, right? It's a very local library mutating around the specific wild type sequence. And we can then evaluate how does this model perform on this library, where here it performs very well, same as the standard model that I described to you before. But now, just like I told you when I described the problem and what motivated us here, we can look at how does this model perform on this completely random data set. So what we did is we created a promoter that has 36 base pairs in the middle of it that are completely random. At each position, at each of the 36 positions, there is exactly 25% chance that you will have either A, T, G, or C. So there is no reference to some wild type sequence anymore. It's completely random sequences. And what we did is we evaluated our model that accounts for this more, let's say, more detailed and nuanced understanding of how promoters function. And what we found is that compared to the best existing model, which doesn't do that, we can, sorry, I'm showing this wrong way around. So this is the performance, sorry about that, the wrong way around. So this is the performance of our model on this completely random data set, right? And it's actually exactly comparable. Sorry, I used the wrong plot here, but doesn't show the contrast between performance on the orange data set and performance on the green data set. But you'll have to trust me that the performance of our model that's trained only on the orange data set is extremely high compared to the model that doesn't account for all of those additional mechanisms of promoter function when applied to completely random data. And what that meant is that the issue with the original kind of standard view of bacterial promoters was that it wasn't accounting for how promoters properly functioned. Because once we did that, we could predict expression levels from random sequences with much higher accuracy than was possible before. And what's really important to point out here is that these models are not based on machine learning, so we understand they're mechanistic models where we model every particular mechanism of how the system works specifically, right? And this gives us a lot of ways in which we can then use those models once they perform well and actually learn new biology. In particular, what we wanted to learn is to use those models to explore their consequences on how promoters can evolve. And so the first, here I'm going to give you a brief highlights of what we found out, because it's kind of interesting and I think a bit counterintuitive if you ever thought about what mutations do. So one thing what we looked at is the likelihood of evolving a promoter, a constitutive promoter, so sigma 70 constitutive promoter from completely random sequences. And what we find is that something between 10 and 20% of all random sequences already are promoters, right? Already are constitutive sigma 70 binding promoters. And here what you're seeing is a measurement of fluorescence and the PDF, a probability distribution function of those fluorescences across all mutants that we measured. And we have an experimental data set, which is the dotted line, and the model prediction, which you can see fits here very nicely. And for sequences that are 36 base pair long, like experimental data, around 10% of all of those sequences already have fluorescence. They're already promoters. But then let's try to think about what happens to the other 90% that are not already promoters. And here in this somewhat complicated plot, what you're looking at is you're looking at the predicted expression of the completely randomly generated sequence along the x-axis, which is split into those sequences that have that are already promoters. So that's the 10% here, and the 90% that are not already promoters. And then on the y-axis, you're looking at a single point mutation in each of these original random sequences. So a single point mutation that has the biggest effect on increasing the expression levels predicted by our model, right? So on the y-axis, you have mutants that starting and with the single point mutation, there's no single point mutation that leads to their expression, which is this area here. And this area here are the mutants that start with no expression, so random sequences that start with no expression, and then you can find a single, at least one mutation that gives them what we call measurable expression we can measure it in a plate reader. And it turns out that from this whole group, which is 90% of all random sequences, 90% of them are in this category, the shaded area, which is the area that tells you that 90% of non-expressing random sequences, you can find at least one point mutation that gives you expression. And this is pretty crazy, right? This means that you're basically generating promoters all the time, everywhere. So the E. coli genome, because we're studying E. coli here, has to do something about that. And what we can then look is there evidence of selection against promoter sequences in the E. coli genome? And within coding regions, and this has been shown before, there's extensive evidence for selection against binding sites for RNA polymerase in the coding region. And so what you're looking at here in this kind of teal color is the predicted number, is the number of sites in the E. coli genome, and the gray is the predicted number of sites based on randomized and shuffled version of the E. coli genome. And what you can see is that if you reshuffle the E. coli genome, you predict to have way more sequences, binding sites for sigma 70 RNA polymerase than you actually observe in the genome. But what's really crazy is that when we looked at only at known promoter sequences, so sequences that are known to have promoter function, even in them, there is statistically significant selection against having too many RNA polymerase binding sites. So it's so easy to evolve binding sites for sigma 70 RNA polymerase from random sequences that E. coli genome has selected against having too many sigma 70 RNA polymerase binding sites specifically in sequences that want to have those binding sites. That's how easy it is to do this. And so really what I wanted to get at here is, so that's what you can learn adopting this kind of mechanistic approach and view of evolution. You can start modeling and predicting evolution in a way that wasn't capable or possible before. And so using this, what my goal here today was to really tell you how we can accurately predict expression levels for any random sequence because we understood the mechanisms of how promoter function better using this large-scale mutagenesis data. And you will learn some lessons, incorporated them. We learned something new about mechanisms of how promoters function, incorporated them into a biophysical framework, and use that to predict the effects of mutations, which then allows us to study evolution in a way that's very difficult to do if you just have to rely on experiments to tell you what mutations do. So that's all, that was a big kind of explanation of the framework and how we think. And these are papers, this paper came out at the beginning of 2022. But what I really want to tell you briefly at the end here is, cool, but that's still pretty esoteric and there's no mention of antibiotics here at all, which is a sneaky thing on my end. So what I want to tell you about a little bit about what we're doing now in my lab. And so my lab is still new, and COVID is and was certainly a thing. So we are, maybe not as far as one could have imagined, but nevertheless, let me tell you a bit what we're doing right here. So we look at a slightly more complex system than what I described to you with constitutive promoters, and we look at a system where we have a promoter that controls the expression not only of a fluorescence marker, but also of tetracycline of the tetase pump. This is an antibiotic efflux pump. And antibiotic efflux is one of the major mechanisms of resistance, that bacteria can acquire point mutations in the target site of the antibiotic. They can find ways to degrade the antibiotic. They can acquire plasmids or horizontally transferred genes that degrade the antibiotic, or they can tinker with the pump that excretes that antibiotic out of the cell. And most of the time, resistance that occurs through the multi-drug efflux pump doesn't come from changes in the pump themselves. It comes from the changes in the regulation. So you change the expression of the pump to allow you to get rid of more antibiotic. And you can see how the kind of stuff that I just described to you can be very relevant for understanding the evolution of such systems. And that's exactly what we're doing by using, again, this very simple synthetic system that allows us now to track how promoter mutations change expression levels, but also how they affect the fitness of an organism. And so to illustrate what I mean, we can imagine some wild-type promoter and its mutant promoter, and the consequences of these are to change the expression of the pump, whereby the wild-type promoter has expression of 3 pumps, those are very fictional numbers, while the mutant promoter increases that to 8 pumps in this illustrative example. And how those organisms behave, how the mutant and the wild type behave, is going to depend on the environment they're in. So if they find themselves in the presence of in the absence of an antibiotic, maybe the wild type promoter is going to be happier because it doesn't have to make all of this extra stuff, all of these extra pumps, which get embedded into the membrane and can cause all sorts of fitness effects. But when in the presence of antibiotic, the mutant might be happier. And here at the very end, I'm just going to very briefly run through a variety of projects that are going on in my lab, and just to highlight various people who are here with me. So we're looking at fitness landscapes of promoters and really studying them in detail. Do they have single peak? Do they have multiple peaks? And all in relation to this biophysical modeling and thermodynamic modeling. And this work is carried out by my postdoc Stepan, here shown on the left, and a few master students, Jeremy and Simon, who were in the lab last year. We also look at the relationship. We also try to study gene regulatory network evolution, but instead of approaching it from this global perspective, looking at the entire network, we try to understand how individual components and the biophysical thermodynamic properties of how promoters work actually give rise to the networks that we see. And this we collaborate with Claudia Igla, who is actually been a postdoc, but she's going to join us in Manchester as a PI in 2023. And then a few other people in Hebrew University of Jerusalem, and a few other people, Magnus and Tobias, here in Manchester. Then we're really looking at predictive genotype-phenotype fitness mapping that extends beyond just looking at single and simple systems like the antibiotic resistance pump, and looks at entire metabolic networks to give us a much broader scope of how we can apply this thinking and this relationship with promoters and their fitness level of organisms. We also work with synthetic biology to help them optimize, to use the kind of approaches that we have to help them optimize synthetic circuits. And then we are focused on multi-drug resistance evolution, particularly, I forgot to outline people, so I apologize. This work is done by Stepan and Ante Maria, a PhD student in my lab. Here it's a postdoc Zahra on the right, and same student Ante Maria. Sorry about that, it's rude of me. And so finally, we have some projects working on multi-drug resistance evolution when one of the drugs affects RNA polymerase, and those are called rifampicin. We're trying to understand how the mutations in rifampicin affect gene expression level in the whole cell, and how that leads to changes in their ability to resist secondary antibiotics. And this work is done by a PhD student, Jake, here on the left, and two master students, Matt and Pat, and Matt will hopefully be joining us as PhD student in the coming months. And finally, as I was telling before, we were looking at the regulation of multi-drug efflux pumps, currently focusing on the simplest one, but we want to extend this to more complex regulatory pumps like ACRI, VTOL-C. And again, this is done by Zahra, a PhD student, a postdoc in my lab, and Rosie, who was a computational master student last year. And so with that, I would like to really thank you. I hope that this wasn't too basic, wasn't too trivial, and gave you some fun ways and some ways of thinking about how we can maybe approach studying evolution in different ways that really uses this, it really leans in heavily on the power we get from relating molecular mechanisms to their evolutionary consequences. So thank you a lot for your time, and yeah, I'll be very happy if there's any questions.
Thank you, Mato. We indeed have already some questions. So the first one would be, what is the greatest challenge in predicting multi-drug resistance evolution?
Look, let me be honest here, there's a difference between a title and what in reality you end up doing, as anybody who does science, I think, knows. If you read our title, you might be misled to think that we are trying to, that somehow at the end of this process, we're going to predict everything all the time. No, we are focused on some specific systems. One of the biggest challenges, in my opinion, is the complexity. There's many antibiotics and there's way more bacteria out there. And understanding the relationship, whatever we learn for a specific organism, here we're studying E. coli. If the RNA polymerase sigma 70 changes and has different binding preferences in another organism, we have to understand those before we can apply the tools that we develop in E. coli. It just so happens that those are very well conserved molecules, so we can, in our specific case, make an argument about their generalizability, at least to some extent. But the issue is that these are extreme, biology is extremely complicated and complex, and there needs to be a lot of people working on these problems to give us any hope of being able to and approaching them from various different angles, And what we do is really applicable to one specific mechanism of resistance, but a lot of other mechanisms of resistance don't really have much to do with regulation, like changing the target site of, let's say, some specific antibiotic. How that works requires very different approaches. But what I would say, and what I'm trying to argue, and why I structure my talk the way I did, is that irrespective of how complex and daunting the problem is, I do genuinely believe that we need to spend way more effort into really nailing the relationship between how systems work and how they can change, as opposed to simply looking at a few outcomes of evolution and understanding, well, this mutation did that and this mutation did this. I'm not saying that's not important. I'm saying those are really important stepping blocks to understand more holistically how systems work. So, but that's my argument, right? Obviously, I would say that because that's how we do things.
Thank you, Mato. We have time maybe for one or two questions more. Another one would be coming from one person. You mentioned at the beginning that your approach is not typical to how the field tackles this problem. Can you elaborate on some of the differences?
Yeah, I mean, it's what I think I just alluded to it a moment ago. It's typically, if you look at what's called fields of molecular evolution or experimental evolution in particular, these are fields that use bacteria most of the time to study evolutionary processes, because bacteria offer some good, they're good model organisms. They divide fast, they can evolve in real time, time frames that are relevant for us humans. And most of the time, what these studies do is look at some, they evolve. They take identical cells, put them in different environments, and track how mutations emerge and which mutations emerge and what they do, how they improve fitness. But it's very difficult from that data to discern why those mutations are the ones that give you the fitness benefit in the first place, right? And that's the difference, is that what we're trying to do is get to that step as to why, not one mutation did something, but why could that mutation even do something and other mutations don't? In other words, and to borrow from Jacob, who published some papers, more philosophical papers that inspired a lot of my thinking, is that a lot of evolution is focused on studying the actual, which is what we see, the consequence of evolution, the actual reality, and looking at what is possible, what possible realities can be discerned. That's what the experimental evolution approach does. It involves many bacteria in the same environment to see what are the possible outcomes of evolution. And what we try to do is delineate between the possible outcomes and the ones that are not possible. And that requires understanding how biological system works, how they work, I think. Or it requires an unbelievable amount of mutagenesis data, but that seems less efficient and although good from Azenta, I guess, but not realistic.
Thank you, Mato. The last question, I believe, for today. In your genotype-phenotype fitness mapping, what happens as generations cycle and you continue to predict expression? If a time element is added, are you able to model the shifts?
Yeah, I mean, great question. Who knows, right? I'll let you know as soon as we do those experiments. The big part of what we want to do is so that we can say, okay, out of the 10 million possible mutations in this promoter, this subset of 3% are going to improve fitness. And then we want to evolve those organisms when we can then test, is those mutations in fact the ones that we see? But the big difficulty is that other mechanisms might exist in the cell that can also mutate and contribute to resistance. And those are obviously not something we're explicitly trying to model or predict. So it's very difficult to answer your question without those experiments. But get back in a year or two, and I will hopefully have a much better answer.
Thank you so much, Mato. I believe that's all we have time for today. I would like to, and we would like to thank you very much for this time, and also to all attendees for joining us today. If you'd like to view this webinar again or share it with your colleagues, a recording will be available shortly. Thank you very much.
Mention this. Can I just say one thing? Sorry, I'm really awful at this, but I do always forget to mention funders. So Wellcome Trust and Royal Society are funders of my current work and the work that's going on in my lab. In the past, it was also funded by ERC. So I apologize for that.
Thank you, Mato.
Sometimes to say this.
Thank you, and I wish everyone a great afternoon.